for some types of vaccines, a virus is also used as a vector?
Yes, exactly. Yes. In fact, some of the strategies for coronavirus vaccine is actually taking a DNA virus called adenovirus and taking out its bad genes, or adeno-associated virus, taking its bad genes out and inserting the spike protein. That's the protein, the key protein from the virus into that, and then infecting people with that virus. That's an approach that's being taken. Yes, it's adenovirus and adeno-associated viruses are the two viruses that are being used in that way to make vaccines. Um, there are other approaches. There are other approaches by using another group is trying to use the vaccinia virus, which is the virus that was used, for smallpox vaccination, and putting this spike protein or proteins from coronavirus into it.
What does that mean exactly? If you put the spike protein into this new vaccine,
Yes.
what is that? Then what happens?
What happens is you get a small infection, a mild infection at the area of injection, and the infected cells will the virus will produce this protein of the coronavirus along with its own proteins, and your immune system will recognize it. So, in a sense, the vector is kind of a stimulant for the immune system to recognize the proteins that you want to have a vaccine to.
Okay, so the virus that we're using as like the vector.
Yes.
It's been prepared in such a way that it's not going to cause the negative effects that SARS-CoV-2, but by having the protein that's that spike protein, it does get an antibody response from us. It's a way to train our body to defend against that specific protein without it being attached to that specific virus, the SARS-CoV. Okay, okay. So then we're prepared, and then if we get infected by SARS-CoV-2, we're producing antibodies that are going right to block the spike protein.
We hope so.
Interesting.